This video is the second in our series about color vision. It is about color matching. In the first video, we introduced the basics of color. We talked about additive and subtractive color creation. In this video, we explore color matching, which is just as it sounds, using three basic colors to create a match to all other colors. This process is important because it is the basis for color science and helps us understand how our color vision works. Let us begin with a brief review. Technically, light consists of electromagnetic waves from a small region in the spectrum that are detected by the eye and interpreted by the brain. The wavelengths of light we can see range from just under 400 nanometers to just over 700. In choosing a particular color, we could take it directly from the spectrum, that would be called a spectral color, or we could create a new color by mixing other colors. Mixing colored light works by the additive mechanism. As an example of additive color creation, let's take the color yellow. Now we could select yellow from the spectrum as a pure color at about 580 nanometers, or we could create a yellow color to match the spectral yellow by making a combination of red and green. Adding colors together to match a specific target color is called color matching. Your eye cannot tell the difference between the pure yellow from the spectrum and the composite yellow from the red-green combination. Color matching is one of the main tools used to investigate how the retina and brain perceive color, and that is where we start this video. Later, color matching will take us into color space, and eventually to a widely used color map called the CIE chromaticity diagram. As you follow along, don't be intimidated by the terms. The general concepts are the important part. Here, we are going to set up a virtual laboratory to show how color matching experiments are done. On the right are three lamps, one red, one green, and one blue. One reason for choosing these colors is because they roughly represent the colors that the cones in the retina are sensitive to. On the left, we have the ability to project any color we wish as a target for a match. For this experiment, we're going to choose the colors of the spectrum, one at a time, which we will project as a test color on the left half of the circle. As our first example, let us continue with yellow from near the middle of the spectrum, wavelength about 580 nanometers. The person making the match can vary the amount of each of the three primary colors projected onto the right half of the circle until it matches the test color. In the original experiments, the colors used to make the match were red at 700 nanometers, green at 546.1, and the blue at 435.8. The amounts of the three colors used to make the match are called tri-stimulus values. 3 colors equals tri-stimulus. You may remember from the first video that equal amounts of green and red light add together to create yellow. This combination creates a match to the pure spectral yellow. Colors that look the same but have different spectral composition are called metameric colors. Color researchers use this strategy to match the whole range of colors from the spectrum. This graph is set up to show the results of the color matching experiments. The location of each of the primaries is shown by the colored dots on the wavelength scale along the bottom. On the vertical axis is the amount of each color required. Here are the results. The color coding shows the amounts of each of the three primary colors needed to match each wavelength of the spectrum. In some ways, this is what you would expect with each color dominating its region of the spectrum. Here is the same information in table form. To continue with our example, the arrow shows where green and red are in equal amounts. It is just about at 580 nanometers where we find yellow in the spectrum. Let's pick another color, red at 650 nanometers. As expected, the color match is almost all red. So far, so good. However, rarely are things that easy. Note in the diagram there is a region where the red line goes below 0 into negative territory. It turns out that not all colors can be matched by red, green, and blue primaries. In this region, a certain amount of red has to be added to the target color before it can be matched by a blue and green mix. Here in the spectrum, you can see that is in the blue-green region. So what does that mean exactly? Here again is our color matching lab, showing the color to be matched is cyan at 500 nanometers. No combination of the red, green, and blue primaries we have chosen can be added together to make this match. So in this case, we have to make a change in our setup. We have to move the red light so that we add some red to the test color. Yes, that's a surprise, but that's how this is done. Adding a bit of red changes the test color, bringing it into a region in which it can now be matched by the appropriate amount of red, of green and blue. The amount of red that had to be added to the test color is considered to be in the negative range. That may be confusing at the moment, but it will make more sense by the end of the video. From color matching, we're going to take the next step, which is a move into color space. The great Scottish scientist James Clerk Maxwell is famous for his monumental main work unifying the theories of electricity and magnetism. One of the other things he studied was color. In this photo he is shown holding a color wheel, the mechanical tool he used to investigate combinations of color. To describe these combinations, he used a mathematical tool called a vector, which is useful in representing a quantity that has more than one dimension. Remember, in our color matching lab we used three primary colors, which were the tri-stimulus values. A color vector is created simply by using these three values to indicate a position on the three-dimensional graph. The three axes, as you might guess, are red, green, and blue. For example, here's a vector extending into color space. The science of color is based on this kind of structure. Here we won't worry about numbers, just the concepts. As an example, think of red, green, and blue color values where 1 equals a fully saturated color. That was 255 in the Paint Color Creator from the last video. If you had a color vector with equal amounts of red and green, but no blue, the values would look like 1, 1, and 0. What color would that represent? It's another way of describing what we saw before. Red plus green makes yellow. Another example would be if you had red, no green, and blue, making 1, 0, 1. The resulting color would be magenta. In between, in the space that fills the box, are contained all the colors that can be created by adding red, green, and blue, which is most of the colors we are capable of seeing. And as a reminder, if you mix equal amounts of red, green, and blue, making a 1, 1, 1 vector, you get white. Now the box that represents our basic color space has three dimensions. It is nice in concept, but awkward in practice. It would be easier to use if we reduce this to 2 dimensions. That's easier to do than you might expect. Here is how it works. We're going to make a plane that goes through 1 unit on each of the axes. It forms an equilateral triangle. Anywhere on this plane the sum of the three values is 1, so this is also called the unit plane. At the end of this section you will see how mapping the tri-stimulus values onto the unit plane is quite a useful way to represent color. Here we are showing the vector for an intermediate color as it pierces the unit plane. Now since this is a plane, only 2 coordinates are required to specify the location where it pierces the plane. We'll come back to that in a minute. You remember our graph of the tri-stimulus colors used to match the spectrum of colors. You also recall there were negative values which, if we wanted to graph them, would fall outside of our color space box. That's inconvenient. We're going to deal with that now, or rather explain how it was dealt with back in 1931. As we have noted, using red, green, and blue primaries cannot encompass all the colors without using negative values. Further, there is no set of 3 real primary colors that can do that job. To solve this problem, a group of very clever people invented an imaginary set of primaries, naming them X, Y, and Z. These are imaginary colors that do not actually exist, but that doesn't matter. Since everything is based on numbers, they could do something called a linear transformation, a way of rescaling the numbers to make the graph behave in a more convenient fashion. Here is the transformed tri-stimulus graph. It looks a lot like the RGB graph, but the negative values are gone. Now, let us map the transformed tri-stimulus values onto a new unit plane, this one based on the x, y, and z values. The transformation was chosen so that the full range of spectral colors now fits into our positive color space, and therefore they all fit on the unit plane. Just as a reminder, we are mapping the tri-stimulus values for the colors of the spectrum. Here we go mapping the vectors in x, y, z space where they pass through the unit plane. A vector near the z-axis would be bluish, toward the y-axis would be greenish, and toward the x-axis would be reddish. As you trace all the vectors corresponding to the spectral color matches, you trace out a line called the spectral locus. In other words, that line corresponds to the colors of the spectrum mapped onto our color diagram. Contained within this border is the full gamut of colors we are capable of seeing. This color map is called a chromaticity diagram. Here is the diagram presented in 2 dimensions. Instead of being an equilateral triangle, it is graphed as a right triangle because it is easier to work with. The position of any color is defined by 2 values, x and y. The third number from the tri-stimulus values is used to represent luminance. Let's take a closer look at the diagram. The line around the outer edge represents the pure spectral colors, with their wavelengths labeled, going from blue on the lower left to green around the top, then yellow, and red on the lower right. The line across the bottom crosses through a range of purple colors, mixtures of blue and red that are not in the spectrum, which is why there are no wavelengths labeled. Here is the chromaticity diagram with all the colors filled in. Contained within the figure is the full gamut of colors that we can see, all neatly specified by 2 numbers, x and y. This model was defined in 1931 by the International Committee on Illuminance. These are the clever people I mentioned before. The initials in French are CIE. The full name of the color map is called the CIE chromaticity diagram. Because there's so much going on with the CIE diagram, we'll cover a few features here. More are covered in a separate video dealing just with the diagram. You remember that white was created by the addition of equal amounts of all colors. In this case, equal amounts occurs at x equals 1/3 and y equals 1/3. This is called equal energy white and is an important reference point. We will look at more whites later on. Specifying color that everyone can agree upon is one of the main purposes of creating this system. Let's pick something practical. The color of red in a stoplight is specified by the Institute of Traffic Engineers to have these color coordinates, putting it in a region of highly saturated spectral red. In fact, here are the entire color specifications for traffic lights showing the allowable range for each color choice. Now, if we want to mix 2 colors, labeled R and G in the diagram, if they are in equal amounts, the resulting color will be at the midpoint of the line connecting the 2 colors. With one in the green region and one in the red, the midpoint color turns out is yellow. The same works for the combinations we showed in the additive color mixing, red plus blue and green plus blue. Another item. Take a point on the spectrum. In this case, take blue at about 470 nanometers. Make a line through white, and where it intersects the spectrum on the opposite side is its complement. In this case, yellow at about 575. Another way to view this is that adding blue and yellow together makes white, a property of additive complementary colors. Now let's go back and look at the primaries that were chosen to create the original color matches. The wavelengths were 700 nanometers for red, 546 for green, and 435 for blue. Connect these dots, and what do you have? The only colors that can be made using these primaries lie within this triangle. Take a moment and digest that thought. Color mixing can only create a new color that lies between the 2 starting colors. which means that red, green, and blue primaries can only make a color inside this triangle. That leaves a significant part of the blue-green region that lies outside the RGB triangle and therefore cannot be matched. Remember in our matching diagram there was a part of the spectrum that required the addition of red to the test color to make the match? Looking at the chromaticity diagram, can you see how that now makes sense? For a color in the blue-green region outside the RGB triangle, say G in the diagram, adding red would bring it back into the RGB triangle where it could be matched by blue plus green. Your color TV and computer monitor also work on the RGB scheme. Before we end, there's one more thing we need to talk about, the color white. The first white we looked at was the theoretically nice equal energy white. One of the interesting things about human color vision is that there is a whole range of colors we are capable of calling white. One way of thinking about this is to consider the range of light bulbs that we call white, from tungsten filament to fluorescent warm white and cool white. The CIE helped deal with this issue by defining a standard set of values for white. When something is heated up enough, it will start to glow with a certain color. There's a physical model that describes the color created at a specific temperature. Probably the most familiar example is the filament in a light bulb. It is made of tungsten and radiates at 2,856 degrees Kelvin. That's called its color temperature. To relate that to the CIE diagram, here are the CIE standard illuminants, points A, B, and C. In other words, this is a range of colors that can all be seen as white. Point A is the color of a tungsten light. Note it is in the region of yellow-orange. Point B is the color value for direct sunlight. Point C was intended to represent average daylight, but it was superseded by a later D series. The most commonly used is D65, equivalent to 6,500 degrees Kelvin, which did a better job of representing average daylight. That's kind of abstract, so here's an example to help visualize those points. This is a photo I took indoors in a museum lit by tungsten light. You get the impression that it has a somewhat yellow cast. As we are obliged not to use flash, and they will not let me carry the painting outdoors, the next best thing is to use my photo software to change the color temperature. This is the photo at CIE standard B as it would look in sunlight. Side by side you can see it is less yellow. And here it is at D65. But when you're standing in the museum, the yellow cast of the first photo is not so apparent. That relates to how your brain perceives color, a phenomenon called color constancy, which we will cover in a later video. In conclusion, the color map, otherwise known as the chromaticity diagram, is a very useful creation. First, it gives us a common language to describe color, and second, the method of its creation from color matching data tells us a lot about our sense of color. We've come a long way in this video, so I think a brief review is in order. We started in our color matching lab. Looking at the colors of the spectrum, we found that we could match most of the colors by combining different amounts of just 3 colors, red, green, and blue. That result is shown in this important graph. Because there was a region of the spectrum that we couldn't match, that required a change in our lab setup. Our new setup involved the somewhat surprising move of shifting the red light so that it added to the test color. Then we could make a match with the remaining blue and green lights. On the color matching graph, that brought the red line into negative territory, which was an inconvenience. Because these are all numbers, some very clever people made a transformation that moved the negative values into positive territory. Taking the values from the color matching data, those same smart people went on to make a color map so that everyone could describe a specific color in a standard way. The mapping process started in 3D color space. Using the color matching values for red, green, and blue was familiar, but had the problem of negative values falling outside the area that was to be used in the map. So, they defined a new set of primary colors that were imaginary, But that didn't matter. It allowed all the colors to fall into positive territory. Mapping the colors of the spectrum in order resulted in a line called the spectral locus. Here is the 2D map of the spectral locus. The bottom boundary includes colors that are not in the spectrum, but can be produced by adding red and blue together. All of the other colors fall inside this boundary outline. In other words, this is the gamut of all the colors that we as humans are capable of seeing. Pretty cool. There is a separate video with more features of the CIE diagram. If you want to read more, here are selected references about color science, and my favorites for general coverage of light, color, and vision.